basically single cell multi-omic measurements can either come from the exact same cell where we do simultaneous measurements of multiple omic profiles, or it can be that we take one biological sample and then for some cells you do one assay as in single cell RNA-seq, and for other cells from the same sample you do another assay like single cell attack. And then you assume that those come from the same cell populations, but they are not exactly measurements from the same exact cells. And this is a nice overview of everything that you can sort of do. Keep in mind, this is already 3 years ago or so there are a lot more probably at the moment. So everything on the right side is where you can do 2 or more assays on the exact same cell. I think practically speaking, the more common and approaches that gives you high throughput, so large numbers of cells and are commercially available relatively easy, are assays that combine chromatin accessibility and RNA. So these are the SciCAR, SNARE-seq, PIRT-seq, and from 10x it's called multi-omic. And the assays combining protein measurements and RNA. So this is CITE-seq and REAP-seq and so on. Yeah, so these are the ones that I think there are already a lot of data available. So probably, in my opinion, if we would develop training for this, these are the 2 assays that we should consider at the moment. Others that are very interesting are when you can do sort of 3 combinations of things like methylation, accessibility, and RNA, for instance, but the throughput is in general lower at the moment, although there are data sets available. So just a very brief comment on why would we actually want to integrate the data, right? As in why wouldn't we analyze each modality separately? I think the most common thing is that we often would want to learn a common embedding of cells, so a representation of cells in a row or dimensional space based on both omic profiles, with the idea that maybe this gives you either higher granularity in which, like in how you split cell types, for instance, or study differentiation trajectories and so on. And once you get to this common embedding space, you can do everything we do on the single cell RNA-seq is probably on the lower dimensional space, right? So you take your single cell RNA-seq data, you do some QC normalization, then you do PCA, and then usually everything happens in this PC space. So you basically build your KNN graph clustering and so on there. So if you get to this common embedding of multiple modalities, then everything that you can do on a unimodal single cell RNA-seq is basically accessible, let's say, right? So you can cluster in that space, you can do trajectory. A lot of people are working on reconstructing noisy data. So if you would measure chromatin accessibility, single cell attack together with mRNA, the accessibility data is way sparser than the mRNA measurements. So you might want to use the mRNA data to guide a little bit imputation or reconstruction of missing data on the attack, or the other way around. I think the third point is probably the most interesting one, right? If you would want to study how things affect each other, like how accessibility at a specific locus affects gene expression. I think this is mechanistically a very interesting problem, but that's probably also the hard part of the analysis, and how can you find relations between features from the multimodal measurement. And the last point is maybe a little bit related, and it's on reconstructing regulatory networks. So if you do this on mRNA, usually you would rely on the co-expression relationships between genes. If you would include accessibility, then you can look into the effects that potential enhancers, for instance, have on gene expression. So there was a nice paper a couple of years ago that sort of split the task a little bit. So if you look at the data matrix, you will have basically groups of cells, right? And then groups of features. And depending on what you have measured, that defines a bit the integration task. So on the left side is basically the batch correction, where you have the same set of features in this sort of unimodal, and you measure that across different groups of cells. So this would be basically that you did single cell RNA-seq or single cell attack, so a unimodal assay on 2 groups of cells, and then you want to do batch correction, right? So you want to remove any differences between the groups. That's basically technical. And that's not what we're talking about today. So what we're talking about today is more the right side of this image, where you can have one of 2 scenarios. So the first one is called vertical integration. So for the same cell, you have multiple measurements. So this would be multi-ohm or CITE-seq data, where the anchor here is the cell. So it's exactly the same cell, and you're measuring multiple things. So yeah, so here I think the idea is that if you try to embed these cells in a low dimensional space based on the RNA profile, and these cells in a low dimensional space based on the attack profile, then these low dimensional manifolds probably align to some extent. And that is the assumption that you can use to integrate the data. I didn't mean to have an exhaustive list of methods, but I just for the vertical integration where you have multiple measurements of the same cell, these are sort of the common tools to do this integration. I think for people working in R, it's mainly the two different versions of integration in SEURAT with the canonical correlation analysis, or more recently, the weighted nearest neighbors. There are also ways to use variational autoencoders, for instance. But across all these methods, the idea is that, again, you get a lower dimensional representation of the cell using both data types that you have measured. And on the other hand, there is way more variable types of methods to do the diagonal integration. Again, the basic trick there, you get an embedding on each data type independently, and then you try to align them. So you assume that in that embedding, for instance, cells were separated by type. And then if that is the case, then you can try to find how they overlap. And this is significantly a more difficult task than the first one. Try to break them a little bit, because the way I was thinking about this is we develop a course or material to train. I always find it difficult not to cover a bit of basics, right? As in, if you're going to say we will use total VI to do this integration, well, it's a model based on variational autoencoders. So you probably need to give an idea what variational autoencoders are. These are sort of the three sort of flavors of methods that people are using in this area. So a lot of methods are doing matrix factorization. So if you would do canonical correlation analysis, NMF, Jive, or whatever, they are all doing some sort of matrix factorization at the end. And I feel this is sort of okay because we do explain PCA in general in the courses. Not deep enough, I guess, but okay, there is a bit of explanation of what PCA does. And from there, you can extrapolate a little bit to other matrix factorization approaches. Goes for the nearest neighbor approach. So there we explain KNN when we get to the clustering step and to dimensionality reduction and so on. So again, the weighted nearest neighbor is sort of an easy extension from that in terms of explanation. Finally, the last part, I think, is a bit more tricky, and maybe that's even going back to the normal single-cell RNA sequencing analysis courses that we give, is that, at least in our course, we don't cover the CPI at the moment, because it just requires a lot of explanations of what variational inference is. But here, I mean, these methods are becoming more and more applied, also in the multi-omic field. So maybe that is something that we want to include. I did look a little bit where are the materials, like who covered actually multi-omic integration before. So these are like 4 links that I found with actual lecture material, from which I borrowed also some of the slides I showed today. So I think the first one was a course taught in France, where there's I think it's a very nice introduction, and then big part of the lecture goes into applying it to cancer application, which I think is quite nice. This is quite famous course on multimodal data integration from ML API, where there is one day actually, if you look at the curriculum, there's one day fully covering single cell multi-omic integration. These lectures is available here by Sheila Zamper, and it's an available lecture, so it's quite nice. And there's the course, the summer school that was organized between NBIS and SIB. There's also there one lecture, and of course the project-based learning aspect of the course. So I think these are already a very, very good start if we want to sort of include this in our teaching courses. I thought a little bit about challenges. I think for now, these are all lectures that fit 1 hour. So you can sort of give an overview. So it's a bit of an extensive version of what I told you now. In my view, we do need to cover a bit more the basics in these methods, because they do make a difference. Even for the matrix factorization, these are very well established methods, but there are things that you should know about how you model your data, different data distributions, for instance, that come from the different omics and so on. So there is a bit of room to make this a little bit more extensive if we want to. I thought about is that in most courses, the first part, the pre-processing part for the single cell RNA-seq is very fast, right? We almost always give the count matrices for people, and that's where we start. And then we give them a very, very short intro on what Cell Ranger does to get you the count matrix for the past, from the past Q files. Maybe you should change this for the single cell RNA-seq because there are also many considerations that you can take into account. Especially with people like we do a lot more long read sequencing now, and then Cell Ranger is completely useless, so you need to do something else. Or if you want to look at alternative polyadenylation sites or so on. And even more important if you have another assay in the data. So if you're working with single cell attack data or single cell ChIP-seq data, then I think it's very important that you know what happened in the initial steps of mapping your reads to get a feature matrix. It also affects a bit the type of QC that you will do and so on. I think the QC on the attack data becomes a little bit more closer to the sequencing reads than what we do on the single cell RNA-seq, where we just quantify total numbers of UMI and gene counts. So perhaps we will need to include a little bit more than that. The thing is that Naveed yesterday mentioned that there are also now protocols coming that can do spatial multi-omics. So you can do site-seq on a piece of tissue, or you can do combined RNA and chromatin accessibility on the tissue, which are really nice, right? I think big part of it is that we will have additional challenges in terms of image registration, for instance. That is something you don't immediately cover, but assume that these assays are done on the exact same slide, then you don't have a problem. But if they are done on 2 consecutive slides, then there is a bit of a problem of how you would align the data. Or if the data comes from different resolutions, as in if you do, I don't know, we do imaging mass cytometry and imaging mass spectroscopy, both have very different resolutions. So how do you basically align your sample, even if they are on the exact same slide? So that is something that maybe together with the spatial course could be mentioned, covered, thought about. I don't know much about this, to be honest. This is the end of my slides. I think my aim here is to think a little bit, do we need a sort of a separate advanced course on multi-omics, or should we think about developing it as an additional lecture in the existing courses? Then the courses also become quite large.
So I would like to just ask, what is your view? Should we do a different course? Because even at MDiS, we also have a course on epigenomics, and then there is like just the basics, like bulk epigenomics, so then ChIP-seq, bisulfite sequencing, and attack-seq. And then there is a little bit lecture on attack-seq plus RNA integration, but it still is also very, very small compared to that. Yeah, so what are your aspirations on that topic?
I think it does deserve a separate course, to be honest, but I would do it also sort of as a more advanced course.
Exactly.
Because in like a similar intro course to what we do now in single cell RNA sequencing, it's probably not will not be very helpful. Right? I mean, you will probably have the tendency to quickly go to a cell by feature matrix and then immediately work with that. And in my opinion, you need to cover a lot before you get to do something actually useful.
But do you think maybe adding, if you have, we have already like a single cell course, if we create one material to, in addition to that, let's also take a data set with attack-seq or a multi-omic if there is available, and do just one lab so that we can kind of add to these repositorial lectures, let's say. At least something short in the beginning, and then we expand that to kind of.
I think we can, but the most practical way would be to only follow the CRAD pipeline because it's relatively fast.
Yeah, exactly.
All the matrix factorization methods are relatively slow to run, also. So you would need to find a way. The WNN from CRAD is relatively fast on small data sets. So that can be. But yeah, I don't know how you would do it in the intro lectures, for instance. So you don't cover single cell attack. So you will need to have a little bit in your intro material about chromatin accessibility in general, right?
Yeah, and then there will be, for example, there needs to be a lecture before that. Yeah, of course.
Yeah, but that's what I mean. Then it.
Yeah. No, yeah. So that's like if we want to add that to like a whole range, we also need to have like one short part and the like much more detail we plan as a different course, right? because we cannot, yeah,
Yeah, exactly. Yeah. On the other hand, I think maybe on the long run, changing the single cell course a little bit will also explain a bit more the basics, right? So then it becomes a lot faster to explain different things on the single cell analysis, but also on the multi-omics
Yeah.
and the spatial and
yeah,
and so on.
definitely, yeah, I agree.